All right, so today we're going to start a series of 3 lectures on immunity, cellular and molecular immunology. We've been talking about cell and molecular biology, and now we're going to focus in a little bit more on immunological processes. So today's lecture, we're going to break it up, and I think Dr. James did a pretty good job of sort of helping to begin to develop the organization, how we organize the immune system. Today we'll talk about the innate immune system and dig into that a little bit. And we're going to do this in the context of a disease known as Crohn's. All right, so what is Crohn's disease? It is a type of inflammatory bowel disease, or IBD. And just as the name implies, this is a disease of your colon and your small intestines, where you have abnormal amounts of inflammation that are often chronic in nature. Okay, it obviously causes a lot of discomfort for those that might suffer from it. It also, if it begins to occur too early in life, it can actually lead to a form of malnourishment or growth retardation. It will certainly generate fever, weight loss, again, because you're unable to absorb nutrients as efficiently as you normally would. If we look at the GI tract, this is a colonoscopy and video, an image from a colonoscopy. What we'll see are these things known as abscesses. That's this funky looking yellow spot here. And essentially what an abscess is a lesion that's filled with dead neutrophils. So it's a pus-filled lesion. So you also get abscesses sometimes when you scab your knee and it gets real pussy. Those are actually that white material that comes out of wounds is oftentimes dead neutrophils. Obviously, it causes a lot of pain and discomfort in your GI tract. It can cause diarrhea, ulcers, as you see sort of here on the right, and maybe that one up there. And if the ulcers continue to develop, it can cause these things called fistulas, where the ulcers literally begin to develop so extensively that they begin to form tunnels and tracts, okay? And so as you can imagine, if I have a tunnel of tissue within my GI tract, it's a place where more bacteria can grow and colonize and accentuate the problem. Other symptoms, anemia, and neurological issues, and we've talked about anemia before. All right, here's the prevalence and incidence numbers. About 1.5 million live with IBD in the United States. That's a lot of us, right? And there are a lot of different factors that go into this, and we'll talk about those here in a minute. Incidence about 1 in 10 cases per 100,000 people in the U.S., and it shows what's known as a bimodal distribution. And what we mean by bimodal is if I were to plot age, the number of cases, right? The number of cases and age, a bimodal distribution is something that'll look like that, right? We have two humps, all right? So what we see is that it disproportionately affects young kids in their teenage years, preteen years, and older folks. And the causes for this bimodal distribution are still not understood. All right, etiology. This is what we would call a polygenic disease. It's not a term that's up here, so this is just, again, trivia facts. It might actually be, you might address polygenic diseases later in the term. Polygenic means it's a result of multiple genes and their involvement in the development of this disease. So you can think about things like diabetes, right? There's not one single genetic mutation that we can point to that says, aha, there it is, you have diabetes. That's to be contrasted with what we would call a monogenic disease. So sickle cell anemia is a monogenic disease. There's one mutation that leads to that disease, right? Cystic fibrosis It's a mutation in one gene, but it's multiple different types of mutations that can occur. Sometimes that's still considered a monogenic disease. Okay. So we know that genetic mutations enhance the susceptibility of patients to causing the underlying innate immune system to malfunction, which is at the heart of Crohn's. And Crohn's develops when the adaptive immune system tries to overcompensate, okay? And we'll talk a little bit about what's happening there. Symptoms are a result of continuous immune response to microbial infections. It's like the immune system, the innate immune system in your colon and your small intestines gets turned on, and it just never gets turned off, right? As a consequence, you not only you're not only attacking foreign pathogens and foreign bacteria, but in our guts right now, hopefully if we're eating a really nice healthy diet, we have what are called commensal populations of bacteria, right? We have what's called a microbiome, right? Everybody's probably heard that term before, in our guts that help maintain digestive health. And so if our immune system gets out of whack, we start attacking those beneficial bacteria as well. All right. We'll talk about a number of engagements of innate immunity in this lecture, but we're going to ultimately end with this gene here, known as NOD2. And this is a gene that's been found over the last decade or so and is associated with both innate immunity and Crohn's disease. And there's been some studies, nice studies to connect the two. All right. So this is a rehash of a slide that Dr. James presented in the pre-lecture. So the important qualities of the immune system or immune response is certainly diversity, right? And he talked a little bit about it, in the pre-lecture, that we have to respond to a wide variety of pathogens. In fact, we have to respond to pathogens that don't even exist right now, that are going to evolve next year, the year after, 10 years from now, 100 years from now. Our system has to be diverse enough in order to tackle these sort of unknown threats. It also has to be specific, right? I can't just target random stuff because I certainly don't want to target myself, right? And the specific responses need to be specific for the type of bacteria or virus or pathogen that we're attacking, okay? So the specificity refers to this idea that we're going to respond to one molecular feature, again in quotes, from a pathogen, and not pathogens lacking that feature, okay? Or ourselves that also lack that feature, critical component of that. All right, and then memory, right? So we need to certainly remember previous pathogens. If we now unpack that and think specifically about innate immunity is the immune system that we all have. We all share that. It's genetically encoded, and you and I are exactly the same for the most part, unless you have a NOD2 mutation, right? Or others. And so this is not a learned. When we talk about memory when it comes to innate immunity, we're talking about memory across evolution, right? So these are features that pathogens consistently display. And so we have evolved very common mechanisms to tackle these pathogens. Yeah. All right. Again, just a rehash of an earlier slide. Innate immunity. We're going to divide both innate and adaptive immunity into two different systems. Those of the blood circulating factors, what we call the humoral immune response, which is a system of enzymes and or other proteins. In this case, we're going to talk about specifically the complement system, which is a system of enzymes that can directly kill foreign cells. Okay? And so we'll talk about how that works here in a minute. The other is that of cell-mediated immunity. And when we're talking about cell-mediated immunity in the case of innate immune responses, we're talking mostly about phagocytosis, right? That's the primary mechanism. What we're going to do is, in this lecture, though, is we're going to expand on that, right? So we have other cellular receptors, other protein systems in our cells that are not triggering phagocytosis, a phagocytic and degradation process of bacteria, but rather are going to engage other molecular mechanisms to help isolate, sequester, prevent further infection. And we will talk about adaptive immunity in the next lecture, so I'm going to not tackle that. All right, so we've got innate and we've got acquired. We're going to focus on innate. And innate is both a humoral response, and we're going to focus today on the complement system as our humoral response of innate immunity. And then we'll tackle the cell-mediated systems. So the complement system is a system of about 35 soluble and cell surface receptors, or cell surface proteins, that are going to detect bacterial pathogens predominantly and directly kill those cellular particles, those cells, okay? There are three main pathways, and they all converge on this protein known as C3. So we'll talk about those three pathways now. So the first is that of a classical pathway. This is a pathway that it's called classical because it's the one that was discovered first, and it's going to utilize your own antibody response. We've not talked about antibody responses regarding immunity, but they're a part of our acquired immune system. So this is one of the points where we have a convergence or an intersection of innate immunity with an acquired immune response, okay? So antibodies are going to come from the acquired immune response. We'll talk about where they come from next lecture. But they're going to bind to this protein C1q. And so when the antibodies are able to detect pathogen surface proteins, right? We get multiple antibodies binding to a bacteria, and that causes the multiple binding events by antibodies on that bacterium will cause the binding or induce the binding of C1q. Okay? All right, so one question, I guess, that can come up is we've talked a little bit about one antibody, the specificity, right? So one antibody should bind one epitope, ideally, on one antigen. So how is it that we can get multiple antibody binding events on a bacterium? Anyone want to fathom a guess? Yeah. Yeah, so that's one option, right? Is the bacteria doesn't just have one protein on its cell surface, right? And so you have multiple proteins. And one of the things that complement utilizes are the Fc domains, which are conserved across all antibodies, right? So we can have multiple antibodies binding to multiple targets on the bacterium. What's another way that we can get multiple antibodies binding to a bacterium? Anybody? All right, there's not just one of every protein on the surface of the bacteria, right? The bacteria has many, many copies of even the same protein on its membrane, okay? So we can even get the same antibody binding to multiple, let's say, protein A's that are displayed on the bacterial surface, okay? All right. So the second pathway is that of lectin, a lectin sensing system. What lectins are sugar-binding proteins. That's the broad definition. In the case of the complement system, we have a specific lectin that is going to bind predominantly the sugar mannose and high mannose content sugars, right? So polymannose types of sugars. And one of the reasons, so that's demonstrated here, right? So we get, again, multiple carbohydrates, these high mannose content containing proteins that are bound by this protein here, and they trigger the complement system. Okay. One of the reasons that they're binding mannose is that bacteria specifically make high mannose sugars. It's just part of their evolution, and we do not, right? So we do have mannose in our sugar structures on proteins that are being displayed on cells, but we don't have this high mannose, these multiple mannose structures. All right, and then the third is, for lack of a better term, alternative. They couldn't figure out how to bend this or call it, name it, so they just name it alternative. And essentially, there are multiple alternative pathways that lead to the activation of this protein called C3. Okay, so what you'll notice about all of these features is that they're all converging. If you look at these arrows, all of these arrows represent proteolytic events. So the complement system is a series of serine proteases that are going to sequentially cleave their downstream target. All right, so we get the activation of, in this case, it happens to be, I think it's C4 and C2. C2 binds to C4, it cleaves, it activates C4. C4 then becomes active and it cleaves C3, right? And so it's through this sequential activation through proteolysis. Our coagulation cascade actually works almost the exact same way. It's a very large cascade of serine proteases that ultimately lead to the activation of blood clotting. Okay? All right. So we're going to gloss over a lot of this, but we want to make sure that it's understood that you have these series of serine proteases that all ultimately lead to the cleavage of this protein called C3 into C3a and C3b. And the key feature about C3b is it's a pore-forming protein. So it's going to form a macromolecular structure that creates effectively a hole in the plasma membrane, in the membrane of that bacteria. You get an osmotic imbalance, and you basically lyse the cell. Okay? So it's a direct cell killing. If I punch a hole in a cell, it will explode, basically, right? I'll destroy it. All right, so this is going to lead to cell lysis. All right, so that's about it for the humoral system for innate immunity. Is this complement-based, pore-forming system in bacteria. Any questions on that? It's pretty straightforward. It's a lot of words to remember, unfortunately. Welcome to immunology. All right, so now we're going to talk a little bit more, and the rest of this lecture is going to be on cell-mediated innate immunity. And how cell-mediated innate immunity works is not only through that phagocytic process that we introduced in pre-lecture, but now more specifically we're going to talk about this pathogen-associated molecular patterns that different pathogens are presenting. And so, and their complementary receptors, or the proteins that are going to recognize those patterns, those molecular patterns. All right, so if we define them, PAMPs, or pathogen-associated molecular patterns, are recurrent features of pathogens that have been targeted by innate immunity. Okay? And this is done over the course of evolution. And this will become a little bit clearer when we talk about the mechanisms that innate immunity is going to use, right? The features on these pathogens, like how can they be conserved, right? Why haven't bacteria or viruses themselves evolved ways to get around our recognition system? If we look at the PRRs, they're defined as host-encoded proteins that recognize PAMPs and trigger the innate immune response. And they'll do it through a number of different mechanisms. I'm going to make a slight little asterisk beside this because I want to be careful. We've talked a lot about being specific with our terminology and being very careful with it. And so I like to point out little incongruities in how we call things versus what maybe they really are. So when we think about PRRs, we're thinking about, it comes from the name pattern recognition receptors. This is a historical designation because, as you can imagine, the first of these that were discovered were all these cell surface receptors that were binding soluble pathogenic patterns. So they were bent as this term of receptor. What you're going to find in this lecture at the end of it is that they're proteins that are not classical receptors that are in this larger family of PRRs. Okay? So in this case, think about this as a historic term and think more about the definition, right, which is correct. Host-encoded proteins. Okay? All right. So they're going to trigger the innate immune response in a number of ways. Chemotaxis is one. So a bacteria activates a macrophage and it causes it to express and secrete factors that are going to say, hey, there's an infection here. Come help, right? Chemotaxis is chemical-mediated migration, right? So it's a chemical factor that's soluble that drives migration up a gradient, okay? The gradient of the expression of that protein. There are anti-pathogen defenses that we'll talk a little bit about. The secretion of cytokines and chemokines, okay? And in this case, I'll make a slight distinction between these two here. Cytokines are proteins that are secreted that are going to trigger additional immune responses. A chemokine is a specific type of cytokine that's specifically going to drive the migration, right, to encourage the migration of other immune cells. That's slightly different from chemotaxis, where you'll see, and in fact, I think Dr. James showed a video of a neutrophil undergoing chemotaxis and following a bacteria, right? So that's a scenario where the bacteria itself is generating something that's a chemotactic factor, right? In the case of a chemokine, that's a protein that's being made by the immune system to call other immune cells to the area, okay? And finally, we'll get changes in gene expression. And we're going to talk a little bit about gene expression in this lecture, even though we haven't actually talked about it yet in class, and so we'll try to put it in context. But ultimately, we want to try to change what proteins are being expressed, right, in order to adapt the cell's phenotype to accommodate or to deal with the pathogen that's present. All right. So we'll run through these. I'll first talk about the classic PRR. And this is calling back. You see none of it's in blue, but it's a really nice place to start because I can point to that really cool movie of a neutrophil chasing a bacteria in a dish. And this is the system that enables that to happen. So one of the classic PRRs is this protein known as formyl peptide receptor, or FPR. And FPRs are actually in the broader class of G-coupled receptors. And we've talked about G-coupled protein receptors before, right? GPCRs, G-protein coupled receptor. And these are going to activate, sorry about that, these are going to activate all those same downstream second messengers, right? PKC, diacylglycerol, IP3, all of those things. And they're going to trigger a polarization of the cell. They're going to increase the activation of cytoskeletal polymerization, and that's going to enable this thing to move. All right, so it's going to activate that PLC pathway. The classic ligand, okay, so here we get to that, what are these repeating structures? What are these repeating factors, right, that we have evolved systems for? In this case, the classic ligand is this formyl methionine, leucine, phenylalanine, this structure here, okay? And it's a very powerful chemoattractant for neutrophils and macrophages. And the interesting thing is, so when a bacteria starts to make a protein, it always starts with a formyl methionine. When we talk about gene transcription and ultimately translation, the start sequence of a genetic structure to trigger the production of a protein is AUG, right? So I always think about school starts in August, right? So that's how I always remember it. And AUG, when it's translated into a protein, from mRNA to protein, that is a methionine. So all translation of protein starts with a methionine. The difference is, in mammalians, we start with a basic methionine. Bacteria use a formyl methionine. And so that evolutionarily is a difference that we've maximized in innate immunity. So this receptor is going to specifically bind this formyl methionine, this formyl group, and it's going to act again as a GPCR and act as a chemoattractant. There are three of these receptor types in humans, and there are actually 6 in mice. Anyone want to fathom a guess why we have 3 and mice seem to be so much more able to handle bacteria? They're in garbage, right? We're not rooting around in garbage, and we haven't been for hopefully quite some time. Mice still are, and so mice encounter far more pathogens than in the bacterial variety than we do, and so they've evolved a much more extensive system. Okay, so that's the classic PRR, and now I want to talk about more relevant ones for us. Okay. So I'm going to talk about this class of transmembrane PRRs known as toll-like receptors. The name toll comes from basically fly geneticists. They like to name their gene names really funny things. They're very creative, unlike those of us in mammalian biology. And what happened was they discovered these in flies, and when they finally discovered the same receptor class in humans, they're close enough to the fly version, so they called them toll-like. Okay? So toll was the original discovery. Humans are very much similar to the original toll, so they're called toll-like receptors. And there's a very large family of them, about 10. Okay? And this was published in 2003, so I can't validate that there are still only 10. There are probably many being discovered today. All right? Toll-like receptors won the Nobel Prize. And they were discovered only about 25 years ago. So these are an important class of receptors. And so this table of TLRs is not in blue, but I have some blue squares here. And that's because what I want you to do is not memorize the TLRs, but to know the different structures and the different repeating motifs that these toll-like receptors are going to recognize, okay? All right, so if we look at classical TLR ligands, they fall into a number of categories. The first 2 are fairly straightforward for us to think about. So toll-like receptor 3 is going to specifically detect double-stranded RNA. We do not have double-stranded RNA in our bodies unless you have an RNA virus. If you've been infected with an RNA virus, in order to replicate, that RNA virus has to replicate its RNA, and in the replication of the RNA, it generates double-stranded RNA. Okay? So in the life cycle of RNA viruses, you generate this product, and toll-like receptors are specifically, toll-like receptor 3 is specifically able to detect that and trigger an immunological response. TLR5, I've played around with this protein a bit, known as flagellin. We talked about flagella on bacteria that enable them to move around, right? That was back in the cytoskeletal lectures. And one of the proteins that are involved in that machine that allows a flagella to wave back and forth is a protein called flagellin. So when bacteria are being degraded by those neutrophils that are releasing all these reactive oxygen species and all these enzymes into the environment, it'll release flagellin, and flagellin acts as a very potent activator of TLR5. These others are, again, a little bit like alphabet soup. LPS, LTA, and CPG. They're designated here. This is LTA, Lipoteichoic acid. LPS is Lipopolysaccharide. We'll talk about that in a second. And the CPG DNA. Okay. So let's dig a little bit into those. And so we'll tackle first LPS. So LPS is, as I mentioned before, is lipopolysaccharide. It's a very specific structure on a very specific type of bacteria. Okay. It has a lipid tail, a very complex lipid tail. It has a core oligosaccharide here in blue that helps hold that lipid moiety together. And lipid A is what helps this thing stick in the plasma membrane, in the membrane of a bacteria. Okay? All right. And then there's this very large glycan polymer. See, it's this structure to the nth, right? This protein or this feature is also known as endotoxin. It gets its name because this is an extremely potent activator of the immune system and leads to apoptosis of many surrounding cells. So you can really trigger huge immune responses by accidentally delivering LPS to a human, right? And so those of us that are in biotechnology and maybe making therapeutic proteins in bacteria, this is one of the primary constraints and concerns the FDA has for that mechanism of production. If we have any LPS in there, I'm just going to induce a huge immunological response in that human. All right, so this is specific to what we call a gram-negative bacteria. And so I'm going to talk real quick, just a slide aside, about what a gram-positive and gram-negative bacteria is. All right, so Graham was a microbiologist, and he's a microbiologist that had determined and defined a methodology to stain bacteria and to subdivide them, right, in order to understand the differences between bacteria. Essentially, there are two different types. There's a type that has two membranes. It has an outer membrane and an inner membrane. And it has what we call the periplasmic space. Okay. What Graham did was he developed a dye that essentially binds a protein called peptidoglycan. And where peptidoglycan is it's sitting on this membrane. Okay. So how this came to be known as gram-negative is this dye that he developed or discovered is cell membrane impermeable, right? So if I add that dye, then its main target, which is on this inner membrane, is inaccessible to the dye. If I look at a gram-positive bacteria, these are bacteria that only have one membrane, okay, with the peptidoglycan on the outside, right? So these guys, gram-positive cells will stain purple, and gram-negative will not stain, or they'll come out like pink, okay? So LPS is our developed way, our evolutionarily developed way to tackle specifically gram-negative bacteria. If we look at LTA, it's a polymer that contains sugars, right? So it's also got a lipid tail, and this happens to be a diacylglycerol linker, right? It's on the outer, it's on the membrane. Has two sugars, and then it has this glycerophosphate chain here, okay? And this is again to the end. So this is a very long polymer chain that's coming off of bacteria. And so LTA is going to be expressed on these gram-positive cells. And so you can see here TLR2 is going to be primed for binding and detecting and responding to gram-positive bacteria. TLR4 is going to be responding to gram-negative bacteria. Okay. So these are things that the bacteria are expressing on their cell membranes. And those receptors on our cells, on our immune cells, are also on the receptor, right? So we're beginning to detect extracellular bacterial stuff, right, in those particular cases. So we're going to shift now, though, to thinking about localization of TLRs and the importance of their localization based on what they're going to detect, okay? And so we're going to focus now on this thing known as cytosine-phosphate-Guanine, or CPG. And CPG is this repetitive two-base stretch. It's a cytosine and a guanine. These are oligonucleotides, right? So these are nucleotides that are part of genomic material that can be bacterial. They can also be viral. And one of the interesting things is you also have this, right? You have these things stretched throughout your genome called CPG islands. We use them for a lot of procedures, including like mapping out where genes are. Okay, but the difference is that in humans and in other mammalians, we have an enzyme known as methyltransferase. And so we're going to methylate this structure. That's this little guy here. So we have that enzyme, but pathogens don't. So there's a slight difference there, right? And so in this case, what it says TLR9 is going to recognize methyl cytosine, and it's going to become activated in response to that. Now, the interesting thing is, right, if I've got a bacteria and it's got non-methylated CPG, right, then how on earth do I detect that? Well, the localization of TLR9 is in the endosome, right? So it's positioned in the endosome. So as a macrophage engulfs a bacteria and begins to start to degrade it, now all of a sudden the DNA is released and TLR4 is sitting there waiting to bind. Okay? So I have receptors on the cell surface to help me. I have receptors on the endosomes to help me. Right? So all along the way, you'll begin to see in a lot of innate immune responses, there's many levels of redundancy and repetition in our ability to detect certain features of pathogens. There are a number of pathogens that we've actually already talked about in this class that have a way of evading normal immune responses, right? And normally they will trigger purposeful endocytosis into the cell, right, as a part of their virulence, right? And this is a way that we've evolved to tackle those bacteria and viruses as well. That they're evading our normal extracellular immune response, and now they get into an endosome, and they're trying to do their thing, and we've evolved a way to try to counteract that. And so you'll find that a lot in immunology. The bugs evolve ways to try to evade, and we, over time, have also evolved ways to counteract those measures, right? And so this is a consistent theme in immunology. All right, we've got a few more minutes, so I want to talk about the last couple, right? So we've talked about cell surface receptors that are capable of detecting bacterial proteins. We've talked now about an endocytic or an endosomal receptor that's able to detect genomic material. And now we're going to talk about some intracellular PRRs. This is where I put the receptors in quotes, right? These are not canonically thought of as receptors, but they're in this family of pattern recognition proteins. The first is this one called CGAS that we'll talk about. And what CGAS is going to do is it's going to actually sense cytoplasmic DNA. All right, so from lecture number 1 or 2, where's all our genomic material? The nucleus, right? So if I have genetic material outside the nucleus, I also actually want to kill that cell because something's really wrong with it, right? So we're going to do that. In the case where we have one of these really tricky bugs that finds its way around our immune system through the endosome and still out into our cytoplasm and is ready to start replicating itself, this is another mechanism that we have. Sort of, you can think about it, the third line of defense, right? So what CGAS is going to do, it's got this really cool structure here, this little coil with what's known as a zinc finger or zinc thumb, right? And what these zinc fingers and zinc thumbs do is they recognize genomic DNA, okay? So it's going to recognize, there's my mouse, it's going to recognize and bind to DNA, and that binding event is going to cause a conformational change, right? And what CGAS is, it's an enzyme, and it's going to make this molecule, this cyclic GAMP. We've talked about the generation of cyclic AMP. You can generate cyclic GMP. What this is it's taking GMP and AMP and it's cyclizing that. Okay? So it's a very specific enzyme that's going to do that. Okay? So it's going to generate this cGAMP, and what that is a second messenger, right? It's a second messenger. It's a product of, in this case, an enzyme that's going to go on and have downstream effects. So cyclic GAMP is going to activate this protein known as STING, right? So here's STING, here's the structure of it. It's going to bind, it causes a conformational change, and what STING does, it leads to the expression of what we call interferon. Okay? Interferon has a number of isotypes or isoforms, right? Interferon gamma is probably one that you'll read a lot about. And what interferon gamma is going to do is a number of things. One is that it's going to be secreted, and it's going to bind to the interferon receptor, and it's going to tell its neighbors, I'm infected, right? So I'm going to be able to propagate information to the surrounding cells, right? And one of the consequences of that is they're going to start shutting down protein translation. So if I shut down protein translation, if I have a virus that's infected the cell, and I stop it from making proteins that are required for viral assembly, now I haven't killed the virus, but I'm trying to prevent its spread. Okay? So that's the main function of interferon. Okay. All right, any questions thus far? It's a lot of terminology. It's a lot of, you gotta love immunology. All right, so here we come. We're not quite gonna be able to finish the lecture, but we're coming all the way back around to NOD 2. So what NOD 1 and 2 are, is they're one of these intracellular PRRs. That protein that we're gonna link to Crohn's disease is one of our pattern recognition receptors. And what do you know? It binds and recognizes peptidoglycan. That same thing that Graham was using as a stain for both gram-negative and gram-positive cells. They both express it, but one is shielded, right? And the other one, it's exposed. And so NOD 1 and 2 are going to bind peptidoglycan. Bacteria make a lot of funny sugars. We talked about high mannose proteins with high mannose structures on them. That's one. This muraminic acid right here is another. And what NOD 2 is specifically going to bind is this muramyl dipeptide. So it's this sugar structure here plus these two peptides, or this small peptide, these two amino acids. Okay? NOD 2 is expressed on monocytes and macrophages and gut epithelium. Okay? So, in general, gram-positive bacteria are not great for our gut, just so you know, right? Because they have this sugar, this dipeptide sugar, presented to our gut epithelium, right? Okay. All right. I don't think we have enough time to get through this. We'll talk about pulling this all together with Crohn's disease on Thursday. Holy cow. On Thursday. Yeah. Okay. And just remember, because Tuesday is a reading day, our next quiz is the following Tuesday, right? We switched from Thursday quizzes to Tuesday quizzes, right? Because of the reading day on Tuesday. So you do not have a quiz on Thursday.